Since the dawn of civilization, there have been many attempts to classify living organisms. It was done instinctively, not using criteria that were scientific, but born out of a need to use organisms for our own use, for food, shelter, and clothing. Aristotle was the earliest to attempt a more scientific basis for classification. He used simple morphological characters to classify plants into trees, shrubs, and herbs. He also divided animals into two groups, those which had red blood and those that did not. In Linnaeus' time, a two-kingdom system of classification with Plantae and Animalia kingdoms was developed that included all plants and animals respectively. This system did not distinguish between the eukaryotes and prokaryotes, unicellular and multicellular organisms, and photosynthetic and non-photosynthetic organisms. Classification of organisms into plants and animals was easily done and was easy to understand, but a large number of organisms did not fall into either category. Hence, the two-kingdom classification used for a long time was found inadequate. Besides gross morphology, a need was also felt for including other characteristics like cell structure, nature of wall, mode of nutrition, habitat, methods of reproduction, evolutionary relationships, etc. Classification systems for living organisms have hence undergone several changes over the time. R. H. Whitaker in 1969 proposed a five-kingdom classification. The kingdoms defined by him were named Monera, Protista, Fungi, Plantae, and Animalia. The main criteria for classification used by him include cell structure, body organization, mode of nutrition, reproduction, and phylogenetic relationships. The three-domain system has also been proposed that divides the kingdom Monera into two domains, leaving the remaining eukaryotic kingdoms in the third domain and thereby a six-kingdom classification. You will learn about this system in detail at higher classes. Let's look at this five-kingdom classification to understand the issues and considerations that influenced the classification system. Earlier, classification systems included bacteria, blue-green algae, fungi, mosses, ferns, gymnosperms, and angiosperms under plants. The character that unified this whole kingdom was that all the organisms included had a cell wall in their cells. This placed together groups which widely differed in other characteristics. It brought together the prokaryotic bacteria and blue-green algae, cyanobacteria, with other groups which were eukaryotic. It also grouped together the unicellular organisms and the multicellular organisms. Say, for example, Chlamydomonas and Spirogyra were placed together under algae. The classification did not differentiate between the heterotrophic group, fungi, and autotrophic green plants, though they also showed a characteristic difference in their walls composition. The fungi had chitin in their walls, while the green plants had a cellulosic cell wall. When such characteristics were considered, the fungi were placed in a separate kingdom, kingdom fungi. All prokaryotic organisms were grouped together under kingdom Monera, and the unicellular eukaryotic organisms were placed in kingdom Protista. Kingdom Protista has brought together Chlamydomonas, Chlorella, earlier placed in algae within plants and both having cell walls, with Paramecium and amoeba, which were earlier placed in the animal kingdom which lack cell wall. It has put together organisms which in earlier classifications were placed in different kingdoms. This happened because the criteria for classification changed. This kind of changes will take place in future too, depending on the improvement in our understanding of characteristics, and evolutionary relationships. Over time, an attempt has been made to evolve a classification system which reflects not only the morphological, physiological, and reproductive similarities but is also phylogenetic, that is based on evolutionary relationships. Kingdom Monera will be covered in this video and other four kingdoms will be covered in separate videos. Let's start with Kingdom Monera now. Bacteria are the sole members of the kingdom Monera. They are the most abundant microorganisms. Bacteria occur almost everywhere. Hundreds of bacteria are present in a handful of soil. They also live in extreme habitats such as hot springs, deserts, snow, and deep oceans where very few other life forms can survive. Many of them live in or on other organisms as parasites. Bacteria are grouped under four categories based on their shape: the spherical coccus, the rod-shaped bacillus, the comma-shaped vibrium, and the spiral-shaped spirillum. Though the bacterial structure is very simple, they are very complex in behavior. Compared to many other organisms, bacteria as a group show the most extensive metabolic diversity. Some of the bacteria are autotrophic, that is, they synthesize their own food from inorganic substrates. They may be photosynthetic autotrophic or chemosynthetic autotrophic. The vast majority of bacteria are heterotrophs, that is, they depend on other organisms or on dead organic matter for food. Next is archaebacteria. These bacteria are special since they live in some of the most harsh habitats, such as extreme salty areas, halophiles, hot springs, thermoacidophiles, and marshy areas, methanogens. Archaebacteria differ from other bacteria in having a different cell wall structure, and this feature is responsible for their survival in extreme conditions. Methanogens are present in the gut of several ruminant animals such as cows and buffaloes, and they are responsible for the production of methane biogas from the dung of these animals. Next is eubacteria. There are thousands of different eubacteria or true bacteria. They are characterized by the presence of a rigid cell wall and, if motile, a flagellum. The cyanobacteria, also referred to as blue-green algae, have chlorophyll A similar to green plants and are photosynthetic autotrophs. The cyanobacteria are unicellular, colonial or filamentous, freshwater or marine or terrestrial algae. The colonies are generally surrounded by gelatinous sheath. They often form blooms in polluted water bodies. Some of these organisms can fix atmospheric nitrogen in specialized cells called heterocysts. Example, nostoc and anabina. Chemosynthetic autotrophic bacteria oxidize various inorganic substances such as nitrates, nitrites, and ammonia and use the released energy for their ATP production. They play a great role in recycling nutrients like nitrogen, phosphorus, iron, and sulfur. Heterotrophic bacteria are most abundant in nature. The majority are important decomposers. Many of them have a significant impact on human affairs. They are helpful in making curd from milk, production of antibiotics, fixing nitrogen in legume roots, etc. Some are pathogens causing damage to human beings, crops, farm animals, and pets. Cholera, typhoid, tetanus, citrus canker are well-known diseases caused by different bacteria. Bacteria reproduce mainly by fission. Sometimes, under unfavorable conditions, they produce spores. They also reproduce by a sort of sexual reproduction by adopting a primitive type of DNA transfer from one bacterium to the other. The mycoplasma are organisms that completely lack a cell wall. They are the smallest living cells known and can survive without oxygen. Many mycoplasma are pathogenic in animals and plants. Other four kingdoms will be explained in separate videos. Thanks for watching. Friends, if you like this video, please subscribe our channel and share with your friends.